Welcome back, DeepVisus explorers, to Where Vision Meets Depth. Today, we delve into the intricate dance of memory and learning, inspired by a groundbreaking paper. In this episode, our journey is guided by the work titled Replay in Deep Learning: Current Approaches and Missing Biological Elements, published by Hayes et al. In May 2021. This paper tackles a critical challenge in artificial intelligence: how to make machines learn continuously without forgetting what they've already mastered, much like our own brains do. How does our brain manage to learn new things without catastrophically forgetting old ones? And can we teach artificial neural networks to emulate this remarkable ability? At the heart of the brain's solution lies a fascinating phenomenon called replay. In biological neural networks, replay is the reactivation of one or more neural patterns, similar to those experienced during past waking events. This process was first observed in biological neural networks during sleep, and it's now thought to play a critical role in memory formation, retrieval, and consolidation. The mammalian brain faces what's known as the stability-plasticity dilemma. This describes the challenge of keeping neural network weights stable enough to preserve past learning, yet plastic enough to acquire new information. Our brains efficiently address this by dividing learning into short-term and long-term memory phases. The hippocampus, a brain structure critical for consolidating short-term memory to long-term memory, is where replay has primarily been observed. Biological replay occurs during various states. It's most notably seen during slow-wave sleep, but also during rapid eye movement, or REM sleep, and even while awake, potentially to facilitate the retrieval of recent memories. Non-rapid eye movement, or NREM sleep, is strongly associated with the consolidation of declarative memory. This is the memory of facts and events. In contrast, REM sleep plays a distinct role, promoting the organization of internal representations, abstraction, and protecting against interference between memories. NREM typically occurs more in the early parts of night sleep, facilitating memory transfer, while REM sleep predominates later in the night, when higher-order memory manipulations occur. Replay in the brain is not an exact duplication of past experiences. Instead, it's often selective and partial, since sleep time is limited compared to the vastness of our experiences. Replaying only selected memories makes consolidation more efficient. What gets selected for replay is influenced by factors like weakly learned information, fear, and reward. Partial replay, where only subparts of selected experiences are replayed, allows relevant memories with shared components to blend, leading to improved generalization. An intriguing aspect of biological replay is reverse replay. When associated with a reward, the temporal order of replay can be reversed, with the strength of this reversal correlating with the reward magnitude. This is thought to be similar to backward planning from a goal in Markov decision processes, a concept widely used in reinforcement learning. Thus, reverse replay could be an efficient way of estimating state values, crucial for reinforcement learning. The precise mechanism for the spontaneous generation of replay during sleep isn't fully understood. However, changes in neuromodulatory tone across the brain during sleep influence neuronal excitability and synaptic strength. Replay during sleep often co-occurs with sleep oscillations, such as sharp wave ripples in the hippocampus and spindles and slow oscillations in the cortex. Coordinated replay between the hippocampus and neocortex has been observed, suggesting these sleep rhythms mediate memory transfer and association across brain regions. Now, let's turn our attention to the world of artificial neural networks. When these networks are trained on non-stationary data, meaning the data changes over time, they often suffer from catastrophic forgetting. This happens because standard training algorithms assume data is independent and identically distributed, an assumption violated in continual learning scenarios. Replay mechanisms have emerged as one of the most effective strategies to overcome this issue. In artificial networks, replay comes in two main forms: partial replay and generative replay. Partial replay involves storing a subset of previous inputs, like raw images, in a replay buffer and then mixing them with new data for training. Initially, many methods replayed raw pixels. However, the field has evolved towards replaying representations or features from the middle or latent layers of a network. This representational replay is more consistent with biological replay, as suggested by hippocampal indexing theory, which posits that the hippocampus stores highly processed representations of past inputs. Generative replay, on the other hand, trains a generative model, such as an autoencoder or a generative adversarial network, to produce samples from previously learned data. This approach avoids explicit data storage, which can reduce storage requirements and address privacy concerns. It's also considered more biologically plausible, as it's unrealistic for the human brain to explicitly store every past input. The selection of which experiences to replay is also a crucial aspect. In reinforcement learning, prioritized experience replay, which replays based on the magnitude of an experience's temporal difference error, has proven more effective and efficient than random selection. Similarly, hindsight experience replay allows experiences to be replayed with various rewards, aiding learning when reward signals are sparse. For supervised learning, while some selective replay methods have shown promise, uniform random sampling often performs surprisingly well, especially for large-scale problems. Despite these advancements, there are still significant gaps between biological and artificial replay. Many artificial implementations differ from how humans learn and assimilate new information. For instance, biological replay happens both independently and concurrently in different brain regions, whereas artificial replay often occurs at a single layer within the network. Furthermore, unlike biology, many artificial replay systems don't purge their memory buffer over time. The distinct roles of REM sleep in abstraction and generalization are largely unexplored in artificial networks. Also, in biological networks, regularization and synaptic changes occur simultaneously with replay, but in artificial networks, these mechanisms have largely been explored independently. Incorporating more biological mechanisms into artificial replay implementations holds immense promise. It could lead to deep networks exhibiting better transfer, abstraction, and generalization capabilities. Conversely, advancing replay in artificial networks can also inform future neuroscientific studies of replay in biology, perhaps by suggesting optimal memory selection strategies. In conclusion, replay stands as a fundamental mechanism for overcoming catastrophic forgetting and enabling continuous knowledge acquisition in both biological and artificial systems. While deep learning has made remarkable strides by adopting replay-inspired methods, significant differences remain between current artificial implementations and their biological counterparts. By continuing to bridge these gaps, we not only enhance the capabilities of artificial intelligence but also gain deeper insights into the astonishing complexities of the human brain. As we close this chapter, remember that the quest for true intelligence, whether biological or artificial, is an unending symphony of discovery. Join us next time on DeepVisus, as we tune into another frequency of the brain, mind, and intelligence, unraveling the mysteries that shape our understanding of thought